Welcome to The Explainer. Today, we're diving into a technique that's, absolutely fundamental in labs all over the world. It's a way for scientists to see the invisible and sort the unsortable. We're talking about electrophoresis.
Okay, so to really wrap our heads around this, let's start with a huge problem that scientists face pretty much every single day. So picture this. You've got this test tube, right? And inside is a super complex mix of all these vital biological molecules. We're talking proteins that make your body work or the DNA that holds your genetic code. You've got to separate them, you've got to identify them, but they're millions of times smaller than anything you can see. So how on earth do you do it?
Well, the solution is, honestly, an incredibly clever technique. You basically make them race. So today we're going to break down exactly how this microscopic marathon works.
Right. So this molecular race is called electrophoresis. And its power really comes from a super simple fundamental principle of physics.
Now that definition might sound a little academic, but really all it means is we're using electricity to push and pull molecules around, but in a really controlled way.
Yeah, the core idea here is something we all learned pretty early on. Opposites attract. When you flip on an electric field, any molecules with a positive charge, we call those cations, they get pulled toward the negative side. And at the same time, any negatively charged molecules, the anions, they zoom toward the positive side. It's really that simple.
But, just like in any race, not all the runners are created equal. Some molecules are way bigger than others, and some have a much stronger electrical charge. And both of these things, size and charge, they totally affect how fast the molecules can move. That's what causes them to separate.
Okay, so if electricity is the force that's pushing all these runners forward, what's the actual racetrack? Well, it's this special gel, and you can think of it like a microscopic obstacle course.
This gel, which scientists have a fancy name for, a support medium, is really just a complex matrix. It's a web full of teeny tiny pores. And the molecules, they have to navigate their way through this tangled mess, which slows them down and, you guessed it, helps separate them even better.
And of course, scientists are smart about this. They pick the right race course for this specific race they're running. So for big, bulky molecules like DNA, they'll use something called an agarose gel. Its pores are pretty big. But for smaller, more agile molecules like proteins, they'll go with a polyacrylamide gel, which has a much tighter mesh. So here's the key takeaway. When the race is on, those smaller molecules can just wiggle and weave their way through that gel obstacle course way faster than the bigger, clunkier ones.
All right, so we've got the principle down. Now let's take a look at how a scientist would actually set this all up and run the race in a real lab.
The whole setup has to be really precise. First, you carefully load your samples into these tiny little pockets, or wells, at one end of the gel. Then, the whole thing gets submerged in a special liquid. It's called a buffer, and its job is to conduct the electricity. Finally, you pop the lid on, turn on the power supply, and they're off. The race has begun. But there's always a catch, isn't there? As that electricity flows through the system, it generates heat, and if you don't control that heat, it can completely destroy the entire experiment.
Yeah, too much heat is a total disaster. For one, it can make your results, those nice sharp bands of molecules, get all blurry and spread out. It can also literally damage, or to use the scientific term, denature, the delicate proteins you're trying to study. And to top it off, it can even create warm and cool spots in the gel, which is like having a racetrack with hills and valleys. It's just not fair.
So that brings us to another big problem, especially when you're working with proteins. See, proteins aren't just different sizes. They also come with their own unique electrical charges. And that would give some of them a totally unfair head start in the race. What we really want is to sort them only by their size.
So how do you fix that? Well, scientists use this really clever method called SDS page. And the secret ingredient is a powerful chemical detergent called, you guessed it, SDS. The absolute genius of this is how it completely levels the playing field. What happens is this SDS chemical, it basically coats all of the proteins. It completely masks their original charges. And instead, it gives every single protein the exact same strong negative charge. So now, the playing field is perfectly level, the race is fair, and the only thing that separates the proteins is their size. Brilliant, right?
All right, so the power's off, the race is over, the molecules are all separated out. But we've got one last, absolutely critical step, actually seeing the results. Because here's the thing, even after all that work, after they're all sorted by size into these neat little lanes in the gel, you still can't see them.
They are completely and totally invisible to the naked eye.
So to see the photo finish, scientists take that gel and they soak it in a special dye. This dye grabs onto the proteins, or the DNA, and just like magic, it reveals this clear pattern of distinct bands. And every single one of those bands, that's a group of molecules that are all the exact same size.
Whether it's identifying the one gene responsible for a disease or matching a suspect's DNA from a crime scene, this powerful yet elegant technique is what makes the invisible visible. It helps us understand the very building blocks of life itself. Which really just leaves one question. What invisible truths will this molecular race help us uncover next? Thanks for tuning in.